The evidence supporting mechanical tension and prompting hypertrophy is pretty much indisputable, but there's more and more evidence supporting the importance of metabolic stress that continues to emerge. Metabolic stress occurs when exercise-induced stress causes an accumulation of metabolites, and this accumulation of metabolites can assist in generating a hypertrophy response a few different ways. Metabolites are simply the pollution produced from strenuous activity. The more you push your limit, the more byproducts you create and the more pollution you produce. If you produce enough, a swollen sensation can occur, which is referred to as the pump. Metabolic stress is maximized during training that heavily relies on anaerobic glycolysis. And anaerobic glycolysis relies on exercises lasting 15 to 120 seconds, or a training program that gives very little recovery. By relying more on anaerobic glycolysis for ATP production or energy, it results in a buildup of metabolites such as lactate, hydrogen ions, inorganic phosphates, creatine, and a few others. It's this buildup of these metabolites that triggers a cascade of circumstances that can create the exercise-induced hypertrophy response. And there are three major contributing factors: muscle fiber recruitment, elevated hormone levels, and cellular swelling. Now, we've spent a lot of time covering motor unit recruitment and how load, velocity, and the mind-muscle connection can aid in increasing muscle fibers used. Metabolic stress increases the recruitment of muscle fibers as well, but when using lighter loads. Let me explain. Recall this slide from the mechanical tension module? Well, let's assume we have to move this car a challenging distance of 1 kilometer. So instead of just pushing it, we now have a distance to move it. You require 3 people to initially move the car. Now, as we reach the 1/3 kilometer distance, one person gets very fatigued and has reached their limit, so they can no longer contribute to pushing the car. And since this person has reached their limit, in order to continue moving it, we have to recruit another person. And now let's say as we reach the 2/3 kilometer mark, another person has reached their limit and can no longer contribute. So we recruit another person to take their place. And this is kind of what's happening with muscle fiber recruitment when it comes to metabolic stress. At the start, you recruit the appropriate number of muscle fibers. But as you fatigue, muscle fibers begin to give out and you start to become weaker. Now, if you push yourself into failure, and that part's very important because you have to really push into the pain, your brain can recruit some larger, initially untapped fibers and bring them into the mix. We don't know exactly why this happens, but it's hypothesized that hydrogen ions accumulate and inhibit the contractility of the muscle fiber. And this contraction failure can then prompt the recruitment of additional muscle fibers. So the key takeaway is when you're training to maximize metabolic stress and trying to utilize motor unit recruitment, you have to train to failure. You have to get your brain to want to recruit the additional motor units. Next, we're going to talk about hormone levels. And this has been an area of great debate over the past few years. It is hypothesized that post-exercise elevation of anabolic hormones via metabolic stress can contribute to muscular hypertrophy. Exercise-induced metabolic stress spikes post-workout hormones, specifically growth hormone, IGF-1, and testosterone. Now, this process heavily relies on the production of lactate, which most of us have experienced at some point or another. Now, the reason this has been an area of great debate is because some of the research shows that this hormonal elevation post-exercise doesn't really do much. However, it's been hypothesized that the primary effect of post-exercise hormonal elevation is to actually increase satellite cell activity as opposed to increasing muscle protein synthesis. Now, if there is truth to this hypothesis, it would mean that this post-exercise hormonal elevation would favor greater long-term increases in muscular hypertrophy without significantly impacting any short-term gains. However, this hypothesis is still fairly young and requires further study. Another thing to factor in is that metabolic stress may influence growth by upregulating anabolic myokines or downregulating catabolic myokines, or both. Myokines have been coined as local growth factors. Muscle tissue directly produces a number of growth-prompting substances. For example, local production of a variant of insulin-like growth factor called mechanogrowth growth factor, or MGF for short, is thought to be particularly important to muscle development, and metabolic stress has been shown to increase its production. Now, the correlation between metabolic stress and local growth factors has not been well studied. Therefore, it's far too soon to draw any sort of conclusions. One of the reasons that post-exercise hormonal elevation has been such a hot topic of debate is because of inconsistencies in the research. And these inconsistencies are often related to demographic factors like sex, age, training experience, nutritional status, and lifestyle. And finally, we're going to talk about cellular swelling, which has been around for ages but seems to have made a recent resurgence. Can you remember that famous Arnold Schwarzenegger scene from Pumping Iron when he talks about the pump sensation? He's referring to cellular swelling. I'm sure you remember pictures like this from high school biology. Osmosis tells us that molecules of a solvent tend to pass through a semi-permeable membrane from a less concentrated solution into a more concentrated solution, thus equalizing the concentrations on each side of the membrane. So let me simplify this a little bit. If we had a solvent like water, and in it we dissolved a given substance, which would be called the solute, we would then have a solution, like the image on the far left. Now, if we divide the solution down the middle with a semi-permeable membrane, like that of a muscle cell, and then add more solute to one side, like in the middle image, then our water solvent would cross through the cell membrane in order to balance out the concentrations, like the image on the far right. And this is how cell swelling works in muscle cells. Nutrients are pulled into the cell and build up during exercise. Increasing solutes inside the muscle is the first way that you could potentially induce sarcoplasmic hypertrophy. Water and nutrients are pulled out of your blood and into the extracellular space between the muscle cells. And then from there, muscle cells pull in more nutrients, to which water follows to regulate the solute concentrations on both sides. And when the muscle cell pulls in this water, it increases its hydration status. And cell swelling is simply a rapid escalation of intracellular hydration. This increase in cell hydration causes the muscle fiber to swell, creating a pumped feeling. So basically, as all these nutrients pull into the muscle, water follows and the cell swells because of all the water it's taken in. The theory is that an increased pressure against the cell membrane from the rapid expansion is perceived as a threat to the cell's integrity. And in response, the muscle fiber is prompted to reinforce the cell structure. If you could cause sarcoplasmic hypertrophy by increasing the amount of solutes inside the fibers, it would be a scenario like the picture on the right. Since water follows solutes, and since muscle fibers are permeable to water, if you add more stuff inside the fiber, water would follow, and voila, sarcoplasmic hypertrophy. Unfortunately, that's not feasible for non-protein solutes. Ion concentrations like sodium, potassium, bicarbonate, calcium, hydrogen, etc. won't change too much in the long term as long as you have healthy kidneys. Now, you also have some fatty acids stored in muscle fibers, and these lipid droplets can grow and divide with training, specifically aerobic training, but they don't attract much water, and they make up such a small fraction of the space inside a muscle fiber that there's really no way they could make much of a difference. So finally, you have glycogen, and glycogen storage capacity can increase with training. However, maximal glycogen concentrations can increase with just about any type of training. They may just increase a bit more with aerobic training or the type of lifting people think will cause sarcoplasmic hypertrophy, like lighter, high-volume bodybuilder style training. Ultimately, glycogen concentrations simply can't make a huge difference either. Even when you infuse glucose and insulin directly into a muscle for 8 hours, glycogen concentrations peak at about 4 grams of glycogen per 100 grams of muscle. 1 gram of muscle glycogen is stored with 3 grams of water. So at most, muscle glycogen plus the water associated with it makes up about 16% of a muscle's mass. Okay, see if you can follow me here for a minute. Average skeletal muscle glycogen concentrations are on average 1.5 to 2 grams of glycogen per 100 grams of muscle. And when they're maxed out, it's 4 grams. So in other words, going from average glycogen concentrations to 100% maxed out, so going from 1.5 to 2 grams to 4 grams, would increase the total muscle mass, which is 100 grams, by roughly 6 to 8%. And that increase would be pure sarcoplasmic hypertrophy. Now, a 6 to 8% increase isn't really a huge increase. Certainly not as large of an increase as what most people expect, given the reputation of sarcoplasmic hypertrophy. However, it's still an important piece to the puzzle, and it still contributes. Now, blood flow restriction training, BFR abbreviated, involves restricting inflow of blood via the use of a pressure cuff. You do this while training with light weights, thereby causing ischemia, which means oxygen deprivation, as your muscle is contracting. And this restriction of oxygen limits slow twitch fibers, thus increasing the recruitment of large muscle fibers. So let's assume you're working with a weight at 50% of your 1RM. When you apply blood flow restriction, your oxygen-dependent fibers, meaning your slow twitch fibers, will fatigue much faster. And remember, normally they're fatigue resistant because of their blood flow, which gives them a constant inflow of oxygen. As they fatigue, your body is compelled to recruit additional motor units to continue the movement. This makes blood flow restriction an interesting and potential future tool because you have the ability to recruit high threshold motor units with lighter loads. And this would spare your nervous system from the demand of heavy lifting. Now, let's review the key takeaways for metabolic stress. The first is to train to failure to maximize motor unit recruitment. And it's really important that you remember it's to train to failure. There has to be a little bit of pain with this one. The second is to maximize your hormonal response and utilize cell swelling. You must accumulate lactic acid during your workout. This requires that you focus on glycolysis and increase your time under tension or adjust your rest time in between exercises accordingly.